In this video, we'll talk about DNA structure in a very basic way. DNA is made out of nucleotides, and so this picture here is showing you what an individual nucleotide would look like. So for instance, this area here is all one nucleotide, and then this is another nucleotide here, and so on and so forth. You'll see that the nucleotides all have three major components. There's the deoxyribose, which is the sugar shown in blue. It's got an oxygen and then 5 carbons. 1, 2, 3, 4, 5. Attached to the 5th carbon is a phosphate. That's a PO4. Here's our phosphate with 1 double bonded oxygen, 1 oxygen with a negative. And on the 1st carbon is a nitrogenous base. And so this is C, which is representing cytosine, which we'll talk about later. Attached to the 3' carbon, or the 3rd carbon of deoxyribose, is a OH. And that basically makes up your single nucleotide. Now, if you want to bond two nucleotides together, you need to make a phosphodiester bond. And that's a bond between the phosphate of one nucleotide and the 3rd carbon of another nucleotide. So you can see that these two nucleotides are bonded together with the phosphate of one connecting to the 3rd carbon of the other. In doing so, the OH group is removed. And so you can see these 4 nucleotides are all bonded together from phosphate to carbon, phosphate to carbon, phosphate to carbon. And so DNA is said to have a sugar-phosphate backbone. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Now, on the other strand of DNA is the same types of nucleotides, but you'll notice immediately that their orientation is different. So DNA is a double-stranded molecule. So this single strand is bonded to this 2nd strand. But in order for that to happen, the strands have to be anti-parallel. And that means that while they're lined up in parallel, one is going in the opposite direction. So if you take a look at the oxygens here in the deoxyribose, they're pointed up in this strand, but they're all pointed down in this strand. Another way to look at that is that in this strand, the top carbon here would be the 3rd carbon. Again, that numbering goes clockwise from the oxygen, so it's 1, 2, 3, 4, 5. So the top carbon here is the 3, and the bottom being the 5, or you can even say the 3, and then all the way down here would be the 5. On this strand, it would start with 5, and the bottom one would be the 3. And so this is what it means for DNA to be anti-parallel. To bond together, the strands have to be going in opposite directions. And the way that they bond together is through the nitrogenous bases. So in DNA, there are 4 nitrogenous bases: cytosine, guanine, adenine, and thymine, usually represented by their letters C, G, A, and T. You can tell over here in this picture that guanine and adenine have a somewhat different structure than thymine and cytosine. And again, you can see that in this picture. In addition to this ring here, there's a 2nd ring attached. So they are both a separate set of bases called pyrimidines. I'm sorry, those guys are the purines. These guys, thymine and cytosine, are the pyrimidines. You'll also notice that in this picture, A and T are always lined up across from each other, and C and G are always lined up across from each other. That's actually really important in how 2 strands of DNA would bond to become a double-stranded molecule of DNA. Because of the polarity of the bonds in each base, there are hydrogen bonds that form between adenine and thymine and cytosine and guanine. And you can see that the slightly positive and slightly negative parts of both molecules line up so that they'll both form hydrogen bonds, which are intermolecular bonds. That means no electrons are shared. It's simply a slightly positive and slightly negative being attracted to each other. And the same is true in cytosine and guanine, this with 3 hydrogen bonds as opposed to the 2 in adenine and thymine. When 2 strands of DNA come together, they take on the shape of a double helix, as shown here and here. And that is your basic structure of DNA.